Mucosa-associated lymphoid tissue is located in anatomically defined microcompartments throughout the gut. The mucosa-associated lymphoid tissues lining the gut are known as gut-associated lymphoid tissue, or GALT. The tonsils and adenoids form a ring, known as Waldeyer's ring, at the back of the mouth, at the entrance of the gut and airways. They represent large aggregates of mucosal lymphoid tissue, which often become extremely enlarged in childhood because of recurrent infections, and which in the past were victims of a vogue for surgical removal. A reduced immunoglobulin A response to oral polio vaccination has been seen in individuals who have had their tonsils and adenoids removed, which illustrates the importance of this subcompartment of the mucosal immune system. The other principal sites within the gut mucosal immune system for the induction of immune responses are the Peyer's patches of the small intestine, the appendix, which is another frequent victim of the surgeon's knife, and solitary lymphoid follicles of the large intestine and rectum. Peyer's patches are an extremely important site for the induction of immune responses in the small intestine and have a distinctive structure forming dome-like structures extending into the lumen of the intestine. The overlying layer of follicle-associated epithelium of the Peyer's patches contains specialized epithelial cells. These have microfolds on their luminal surface instead of the microvilli present on the absorptive epithelial cells of the intestine, and are known as microfold cells, or M cells. They are much less prominent than the absorptive gut epithelial cells, known as enterocytes, and form a membrane overlying the lymphoid tissue within the Peyer's patch. M cells lack a thick surface glycocalyx and do not secrete mucus. Hence, they are adapted to interact directly with molecules and particles within the lumen of the gut. M cells take up molecules and particles from the gut lumen by endocytosis or phagocytosis. This material is then transported through the interior of the cells and vesicles to the basal cell membrane, where it is released into the extracellular space. This process is known as transcytosis. At their basal surface, the cell membrane of M cells is extensively folded around underlying lymphocytes and antigen-presenting cells, which take up the transported material released from the M cells and process it for antigen presentation. And that's mucosa-associated lymphoid tissue is located in anatomically defined microcompartments throughout the gut. Have a great day.